Good morning, students. Today we are going to discussing about another important concept is seed. Another important concept is seed. First of all, how to develop the seeds in flower, in plant body. For example, you know that two male gametes are entered into inside of the embryo sac. The first male gamete fuses with the egg cell, that is called syngamy. The resultant structure of the syngamy is diploid zygote. Diploid zygote, it undergoes many number of the divisions and finally developed into diploid embryo. The diploid embryo developed into seed. In this way, inside of the seed, inside of the ovary, the seeds are developed. Here, in angiosperms, the seeds are end product of the sexual reproduction. Definitely, the end product of the sexual reproduction is nothing but the seeds. It is a post-fertilized product. After fertilization, the ovules are converted into seeds. After fertilization, the ovary converted into fruit, the ovules are converted into seeds. Actually, seeds are developed inside of the fruit. The seed is made up of, seed is made up of seed coat and embryo. Seed is made up of seed coat and embryo. Here embryo made up of embryonal axis, fumule, radicle, one cotyledon in monocots, two cotyledon in dicots. Here, seed is made up of seed coat and embryo. The embryo made up of embryonal axis, fumule, radicle, one cotyledon in monocots, two cotyledons in dicots. Here, now we are going to see dicotyledon seed. Dicotyledon seed. What about the characteristic features of dicotyledon seed? Dicotyledon seed. For example, if you see in one dicotyledon seed, here this is one seed. Actually here, this is also known as seed coat. This is totally seed coat. The outermost layer of the seed is called seed coat. The outermost layer of the seed is called seed coat. Seed coat has two layers. The outer layer is, this is totally seed coat. This is outer layer. The outer layer is testa. Inner layer tegmen. Generally, the ovule is protected by two protective wall layers are called integments. The outer integument is The outer protective layer of the ovule is called outer integument or exostome. The inner protective layer of the ovule is called inner integument or called endostome. This is before fertilization. After fertilization, the outer layer of the ovule, outer integument is converted into outer layer of the seed, that is called testa. The inner integument of the ovule converted into inner layer of the seed coat, that is called tegmen. That's why after fertilization, outer integument is converted into testa, inner integument is converted into tegmen. Next one. Here there is a scare called hilum in the seed coat, through which the developing seed attached to fruit. For example, here the small scare. This is actually a small scare, like that. This type of scare is present. There is a scare is called hilum. This is called hilum. In the seed coat. This is totally seed coat, through which the developing seeds were attached to fruit. For example, here this is actually one bean fruit. This is actually fruit. Here these are also nothing but seeds. This is a seed coat. Here this is also nothing but a scare. This is a scare. Here actually there is a scare is called hilum. This is called hilum in the seed coat, through which the developing seed were attached to fruit. That's why this is nothing but stalk of the seed. Here actually one side of the stalk is attached to hilum place. Other side of the stalk is attached to fruit. This is one more important point. Next one. Here near the hilum, a small pore is present. This is called micropyle. Near the hilum, a small pore is present, that is called micropyle. Within the seed coat is the embryo, which consisting of an embryonal axis, fumule, radicle, and cotyledons. For example, this is a seed. Within the seed coat is the embryo. So this one here, this is actually one embryo. For understanding purpose inside of the seed, here I will show the embryo diagram. This is one embryo. Here actually from here to here, this is called embryonal axis. Next one. This is also known as fumule. This is cotyledons. This is the cotyledonary nodes. This is a radicle. Here this is also known as a radicle. That's why within the seed coat is embryo. It consisting of embryonal axis. This is from here to here, from here to here. This is called embryonal axis. Femule, radicle, and cotyledons. Next one that is also known as the cotyledons are. These are nothing but the cotyledons. The cotyledons are fleshy and full of reserved food materials. The cotyledons are fleshy and full of reserved food materials. Here, the cotyledons are hinged. The cotyledons are hinged to the embryonal axis at the point of cotyledary node, at the point of cotyledary node. If you clearly observe the diagram, these are cotyledons. The cotyledons are developed. The cotyledons are hinged to the embryonal axis at the point of cotyledary node. This is nothing but a cotyledary node. At the two ends of the cotyledons, at the two ends of the embryonal axis. This is totally embryonal axis. At the two ends of the embryonal axis, femule and radicle is present. Actually, this is nothing but a femule. One more name is shoot tip, and one more name is first apical bud. First apical bud. Here next one, this is radicle, root tip or embryonic, first embryonic root. That's why at the two ends of the embryonal axis, femule and radicle is present. At the two ends of the embryonal axis, first apical bud and embryonic root is present. During the seed germination time, femule developed into shoot system, radicle developed into root system. How to shoot and root systems are developed from the seed? How to root and shoot systems are developed from the seed? Like that, in this manner. During seed germination time, femule developed into shoot system, radicle developed into root system. Next one. Here, the regions of the embryonal axis. This is from here to here. This is nothing but a region. The regions of the embryonal axis between the radicle and the cotyledary node. Between the radicle and the cotyledary node. Here, hypocotyle is present. Whereas between the femule and the cotyledary node, here this is also known as epicotyle. In this way, inside of the seed, so many parts are developed from the embryo. Here, how to seed is germinated? Within the seed, there is no embryo. How to seed is germinated? Within the seed, mustard embryo is present. From that embryo, all these characters are developed. All these parts are developed. Next, one more point that is also known as cotyledons. These are in fact the leaves of embryos in dicotyledons. in dicotyledons, which are fleshy and store the which are fleshy due to storage of food materials. Food materials, but in monocots, the cotyledons does not store food material. Does not store food material. Here, if you properly observe the dicotyledon seeds and monocotyledon seeds, in dicotyledon seeds, the cotyledons are store the food materials, but in monocotyledon plants, the cotyledons are does not store the food material. Why here? In how to actually in monocotyledon seeds where the food materials are stored. In monocotyledon seeds, the food materials are stored in endosperm. This is dicotyledon seed information. Later. Next one is monocotyledon seed. This is one monocotyledon seed. Generally in monocotyledon seeds, monocotyledon seeds are generally non-endospermic. So generally endospermic. Monocotyledon seeds are generally endospermic. Maximum almost all type of the monocotyledon seeds inside of the seed definitely endosperm is present. That's why this is nothing but endospermic seeds. But as seeds of orchids, non-endospermic. Orchid seeds are monocotyledon seeds, but in orchid seeds, inside of the seed, there is no endosperm. That's why in orchid seeds are non-endospermic. Based on this condition, all monocotyledon seeds are not endospermic, but some monocotyledon seeds are shows the non-endospermic condition. Examples of non-endospermic monocotyledon seed orchids. Next, first of all, here in the seeds of cereals, such as maize. This is one maize seed, such as maize. The seed coat is membranous. This is a seed coat. The seed coat is membranous and generally attached with fruit wall. This type of the condition is absent in a dicotyledon seed. In dicotyledon seeds, the seed coat is not attached to fruit wall, but in monocotyledon seeds, especially maize, the seed coat is membranous, generally attached to fruit wall. Here, this is totally endosperm. Here, inside of the seed, endosperm is present. For example, if you see in dicotyledon seeds, here in dicotyledon plants, the endosperm is completely utilized for the development of the embryo. That's why here that type of the seeds are non-endospermic seeds. If you see in monocotyledon seeds, in monocotyledon plants, the endosperm does not completely utilized for the development of the embryo. some of the endosperm is remained inside of the seed. That's why monocotyledon seeds are nothing but endospermic seeds. This is endosperm. You know that how to develop the endosperm. Endosperm is a post-fertilized product. Endosperm is formed from after triple fusion. After triple fusion, the endosperm is bulky and store the food materials. In dicotyledon seeds, cotyledons are store the food material. In monocotyledon seeds, endosperm store the food materials. Next one. Here this is totally endosperm. This is actually embryo. The outer covering of the endosperm. See this one, this is nothing but the outer covering of the endosperm separate the embryo by a proteinaceous layer that is called aileron layer. Here the outer covering of the endosperm separate the embryo by a proteinaceous layer is called aileron layer. Generally, aileron layer only appears in monocotyledon seeds. The ploidy of the aileron layer is 3N. Why the ploidy of the aileron layer is 3N? Because of the aileron layer developed from the endosperm. Endosperm ploidy is 3N. That's why aileron layer ploidy also 3N. Next one. Here this is also endosperm. This is embryo. The embryo is small, situated in a groove. This is a small groove. Embryo is small and situated in a groove at one end of the endosperm. At one end of the endosperm, maximum most of the seed is occupied by the only endosperm. Okay, here only at one end of the endosperm, the embryo is small, situated in a groove, only at the one end of the endosperm. Next one here, the embryo, it consisting of an embryonal axis, and the embryo consisting of one large shield-shaped cotyledon is also known as a scutellum. Here in monocot, how many cotyledons are present? 1. In monocot seeds, the cotyledon does not store the food material. In monocot seed, endosperm is store the food material. The embryo consisting of one large shield-shaped cotyledon is also known as scutellum. Shield-shaped cotyledon is also known as scutellum. The ploidy of the scutellum is 2N. And including with the scutellum, the embryo, it consisting of an axis like femule, radicle, epicotyle, hypocotyle. The cotyledon developed from at the point of the embryonal axis, that is called cotyledonary node. Why in monocotyledon seed? Here one cotyledon is present. Generally in monocotyledon embryos, two cotyledons are developed. But one cotyledon is present in the form of suppressed condition. That suppressed cotyledon is also known as epiblast. Finally, here this is nothing but a femule. This is nothing but a radicle. The femule protective sheath is also known as coleoptile. This is nothing but a radicle. The radicle is protected by protective sheath is also known as coleorhiza. Coleoptile is nothing but a protective sheath of femule. Coleorhiza is nothing but a protective sheath of radicle. Yeah, this is nothing but here the total information regarding to monocotyledon seed. Here in this video we have discussed the dicotyledon seed and monocotyledon seed. Thank you all of you.